At the ends of the eukaryotic chromosomes, we have telomeres. Telomeres are present at the ends of these linear chromosomes, and what they consist of are just long stretches of short repeating sequence. The reason why we have them is that it helps preserve the integrity and stability of chromosomes. You could think of them kind of like the little plastic caps at the end of your shoelaces. They're going to help keep the DNA helix from unraveling. They're going to protect the ends during this replication process. And each time a cell replicates, each time the DNA replicates, the telomeres get just a little bit shorter. And most cells have a set number of times that they can divide before they basically stop dividing. And we call this process senescence. So the reason why we have them, let's look a little bit more closely. In the lagging strand template versus the leading strand template. Okay, in the leading strand template, everything is fine, right? It goes from the 5' to the 3' direction, everything's great. However, in the lagging strand, remember we have the Okazaki fragments and this discontinuous synthesis. So the RNA polymerase will put down, or the primase will put down these RNA primers, then DNA polymerase will come along and extend this. However, once this RNA primer is removed, at the very end there is no free 3' hydroxyl group from which to elongate. So what telomerase does is it directs the synthesis of the telomere repeat sequence to fill the gap. This is the enzyme with ribonucleoprotein. It has an RNA that serves as the template for the synthesis of its DNA complement. And it does this through reverse transcription. So when this enzyme is present, it can first fill in the gap right here. As you can see, this little primer at the end is removed. This gap right here is then sealed by DNA ligase. So this enzyme, though, is only active in certain types of cells, specifically stem cells and cells that are destined to divide many, many different types of times. This is also very active during development. So like I just said, in most eukaryotic somatic cells, this telomerase is actually not active. And with each successive cell division, telomeres shorten, they're going to erode, and this causes further cell division to stop. However, in stem cells, as well as certain cells do retain the ability to continue to divide, and malignant cells. Malignant cells often maintain this telomerase activity and are thus immortalized. They can keep replicating. Those telomeres are never going to shorten. So what I want you to do is to read through the case study from the end of the chapter on telomeres and on telomerase, and this is on page 289. And what I want you guys to do is to address and answer some of these questions in the discussion forums. For example, thinking about telomeres and telomerase, how might that be applied to different anti-aging strategies? Do you think it's a good idea to try to use this to, say, so-called reactivate some of our cells, and so we don't go through this aging process? Why or why not? Things like people suffering from chronic stress may have more issues with this. So go ahead, read through some of those case studies and questions and answer those in the discussion forums for this week.